do some sort of paperwork here. So number one, I received 31 midterm exams and got things back to you. Thank you very much for putting it in Word, simply because the computer that I have and am using doesn't have Adobe Acrobat, so I can't convert PDF files. They're a pain in the neck. But I got all of your files, and I'm 99.9% sure that every time I received it and logged it in, I sent you a message back saying I received them. Of course, I have not started grading part 2. And why is that? Because so far I have completed 26 of the one-on-one meetings on part 1 with you, and I have the remaining 5 people scheduled for tomorrow. So Nichtenhauser, Esteron, Takarani, Ferdinand, and Lyle will all be taking exams tomorrow. So that will then round us up to 31. I've basically been looking and trying to purge the people who seem to have stopped coming and things like that, and may or may not have been withdrawing yet. If you remember, I think we were at 36 to 37 people, but we had, I think, at a group of 31. Okay, so probably after the 23rd, all of a sudden my Zoom schedule gets much more relieved, and I'll be able to start doing the grading of the part 2 of the exam 1. Okay, just a point. What I'm finding, and again, it may be the length of time that I'm taking to do this and whatever, but even though this is a very large class, and even though this is and it's being done remotely with all of the attendant problems with it, I have found that as an entire class, people working on part 1, you as a class are doing quite significantly better than I would find in all of the other 40 years that I've been doing this. And again, I realize what I always find interesting in this is one thing that you don't have to do, and I'm saying this in a joking manner. I hope no one takes offense. But what is funny is that we do it on a one-on-one basis. So of course that puts a little bit more pressure on you, even though it's remote. But one of the other interesting things is I would find that the vast majority of you would end up being excellent guests on the game Jeopardy, because in many of your cases, when you answer one of my questions, you usually answer it in a questioning manner. So it's the central sulcus, right? So anyway, whatever you're doing, and again, I hope. But on the other hand, this actually is for a lot of you, the vast majority of you are first or second year graduate students. And you're not used to probably oral exams on a one-to-one basis. Well, if you go on into, even with a master's degree, and you have a master's thesis defense, very often you will have a group of 3 people questioning you. So this is probably a good thing or good practice to go and feel. And the other interesting thing, I think one person said, they said, I'm not sure whether I'm excited or nervous. But I think as the exam would go on, whatever nervousness the person was having was moving towards excitement or arousal in terms of doing things. So in any case, that's that for that. And it's going to become more important because when we do part one of the final exam, it will be based on 100 questions, okay? And I'll be going through that as we move along. Okay, so that's the exam. And like I said, I would hope I'd be able to give you all feedback by the time of the next time that we meet. If I can't, I'll just put it off and then give you the feedback. But the way I'm always going to give feedback is I'm going to give it to all of you pretty much all at the same time, obviously privately. Okay, so the other thing that I wanted to talk about today is the paper. So the paper is going to be due right around the point of the last class. What I would typically do is I'd have people handing me in the papers at the point of the final exam where we would meet, and I would then give part one of the final exam, and then I'd sit there while everybody's taking part two, but you're doing that remotely. And it's a somewhat depressing thing because what am I able to do? I'm able to grade your papers while you're taking the final exam and then give it back to you. So in any case, we'll pick a date, but figure around, let's see, the date of the final is December the 3rd. If we need to go to December the 10th, I have no problem with that, but we'll figure this out and we'll talk about it as we get closer. So now, you obviously have read the syllabus and you see that the paper is worth 10%. Now, why the hell do I have you doing the paper? Well, like I said, over 40 years ago, when I started doing this course, virtually all of the students that were in the class were incoming students in their first semester of graduate school. And one of the things that I always feel that is important that a person learn very early in their academic graduate career is to do a task that somebody asks you to do and not necessarily something that you want to do, okay? I'm perfectly at home with you wanting to do X, Y, and Z, and I want to write a paper on this, or I want to write a paper on that, or whatever. But what I basically do is put in some constraints. So the first thing that I'm doing is in some way I might be taking you out of your comfort zone a little bit. The second thing is way back in 1979, we had no such thing as an internet. We had very little things having to do with clunky computers, and you certainly didn't have PDF files, okay, sitting around. So very often, what this was going over to that cavernous building somewhere called a library, and then having to go into the library and go into the journal stacks and basically look up papers and then fight with somebody else to get online to Xerox the paper and whatever. Well, you don't have to do all of that. However, what you have is an enormous proliferation of journals and whatever. So one of the things that you do is you're going to become acquainted with, if you haven't already, various types of search engines. And the first major search engine that I am basically going to tell for 99.99% of you is PubMed. So there may be people that never heard of it. You can go Google and just type in PUBMED, click on it. And basically what it is Library of Congress looking at medical types of papers. So now, once you get into PubMed, now you can go and search. You can start searching for terms. And then what you can basically do is if you type in a term, by the way, you could type in a term and all of a sudden get 294,326 hits. Well, then what you then have to do is start to cut down on your thing. Also, what you can do is you can do settings where you're looking at most recent. So that comes to the top. Why? Because what I would like you to do, it's not a rule, but I want, when you're going to pick 5 papers to review for me in this little paper that you're writing, first of all, the paper that you're doing shouldn't be a review paper. It should be, each of them should be primary source papers, a thing that's doing experiments, okay? So so now what I have is the other constraint that I have is that the paper should be neuroanatomical. So I'm not interested in the structural model of Freud. It's perfectly good thing to go and write a paper on, do it in another course. Okay, not here. So now here comes the very basic definition. Okay. And I have it as a 2x2 choice thing. You know what an independent variable is. An independent variable is something that the person is manipulating. And you know what a dependent variable is. A dependent variable is something that you're measuring. So what would be an ideal ideal set of papers that you would use for this paper? And that is if the independent variable or variables and the dependent variable or variables are both neuroanatomical. Okay? That would be the ideal thing, because that would be a neuroanatomically aligned paper. So in something like this, what you might be able to do, or the study might do, is somehow down-regulate brain site A and then look at some anatomical measure in brain site B, okay? There, the independent variable is anatomical manipulation. The dependent variable is anatomical manipulation. That is that's a perfect type of paper or series of papers that you can pick. What's the second type of paper that you can pick? That is if the dependent variable, the thing you're measuring, is something anatomical, but the independent variable or variables are things that are not anatomical. So, for instance, one of the things that you might be looking at here is the dependent variable is measuring some anatomical measure in some part or parts of the brain, whereas the independent variable can be the sex of the subjects, or it could be that the subjects are from different patient groups, or that they're different inbred strains of mice, or whatever. We can go on and on and on. But you notice there that the dependent variable is going to be something anatomical. So now, what is not a good paper, or in my or not following what I'm asking for? It is a paper in which the independent variable is non-anatomical and the dependent variable is non-anatomical. Okay, so what you're measuring is the effect of age on depth perception in muskrats, okay, or something. By the way, that paper is wonderful. That paper, you should write that paper for a course in perception, not in my course, okay? Because I'm giving you marching orders. Now, the most common mistake is where people do a paper, and there's literally tens of thousands to hundreds of thousands of papers, that because they are the very basis of behavioral neuroscience, in which the independent variable is something anatomical, but the dependent variable is something that's behavioral. So what that can be is we lesion the CA1 field of the hippocampus, and then we look at short-term memory. Again, wonderful topic. Go if you're writing that in some other field or whatever, go for it. Write that all the time, not in this study. What I want you to basically be doing is remembering the dependent variable is anatomical. So, so now that you basically decide what you might like to study, and by the way, many of you, luckily in this sort of 1 hour meeting that I have with you, we have time left over after the exam. And I've been talking to everybody, asking where they came from, where they are now, whether they're in a lab, this, that, and the other thing. What are their interests? What are their goals? You can obviously fashion your paper very much in line with what your interests are and whatever. It just has to have an anatomical measure to it. So what you have to do 5 papers. What's the easiest way to do these papers? The easiest way is to basically have something that you're interested in, and then find a relatively recent paper that's in that field. And it's by the Schmo lab, okay? And usually, what do I mean by the Schmo lab? In many cases in neuroscience, the laboratory from which the paper is coming is usually the author is the last author, okay? Simply because that's the lab the person has, and the first author is either a postdoc or a doctoral student or a master's student or an undergraduate. The lab it came from is usually the last author. So the easiest thing for you to do is, if you have an active laboratory that's been studying this, you can pick 5 papers right from that same laboratory. And what is going to basically happen? That person and that laboratory is going to give you a very strong insight into how they have studied this sort of related problem over 5 papers and over time. So in a lot of ways, the paper almost gets written for you, okay? At the end of the day, you might disagree with the person, and you can write that sort of in the end, but what you're basically doing is you're looking, you're using that. Now, a lot of you are very clinically aligned or interested or whatever. And one of the things that lots of people end up writing on is fMRI studies. And sometimes what they end up doing is picking, I don't know, we pick a particular form of depression, okay? And there's tons of papers out there that write about depression and fMRI, and maybe looking at a particular drug or looking at a particular type of treatment. And then they look at these fMRI measures. Sometimes when you do that, it becomes a much harder paper to write, because let's go to this other extreme, in which you pick 5 different labs doing a study looking at, let's say, fMRI in the frontal cortex, okay? And then what you end up getting is you read the 5 papers, and it's like Goldilocks and the 3 bears. If you remember the story of Goldilocks and the 3 bears, you were in this extreme, you were in this extreme, and then you had just right. And sometimes what you end up with is the 5 papers in which 2 of them find increases, 2 of them find decreases, and the 5th one doesn't find any no effect at all. It becomes much harder to write, because then what you're doing is trying to figure out exactly what were the differences. Were the differences simply because of sampling error across the things, or were there subtle differences between how people measured and this and that and the other thing? So it becomes a harder type of paper to write. Can you write such a paper? Of course you can. But one of the things I'm basically saying in this sort of exercise that I'm having you do is to sort of simplify the thing. Now, the other point that's really important here is, again, you send me emails all of the time. A lot of you have done that. So when you gather up the 5 papers, before you sit down and start, you think these are good candidates simply because probably you've read the abstracts of the papers and they look like they're right on the right track or something. What you can easily do is just send me a PDF of the 5 papers, or a PDF of the 5 abstracts, and then just say, is this okay? And then in the message say what you're basically looking at. I will look at it and I will tell you, does it fall into category 1, category 2, category 3, category 4 of independent variable dependent, and say, go for it, or you might reconsider that. This is totally, you can do that. You can do it, and I will give you feedback, or you don't have to do it. One of the problems, though, I'm going to tell you, is that every so often, it doesn't happen that often, but a perfect person writes a perfectly good paper, and I look at it, and neither the independent variable nor the dependent variable had those anatomical measures. So I end up having to give the person, I write down an excellent paper, but you get a 6 out of 10, simply because you didn't follow the instructions. And the whole point of it is doing something like this. Let's imagine each of yourselves 8 years down the road. You now have the degree that you want, and you're sitting there ready to answer a call for grants, okay? So what happens is there are calls from like NIH or whatever that says, we are encouraging people to submit grants on topic X. And you sit there and you work for weeks, 10 hours a day, refining everything, thinking along the way, and then sending it in. And of course, what you wrote has nothing to do with what the call was. What happens to your grant? Boom, it gets thrown in the waste paper basket. Okay, so you just did an awful lot of work. By the way, very good work, but it wasn't for what the people asked you for. So you basically have to begin to learn that and whatever. And that's what this paper is there for. So do people have any questions on this? It's going to be about a 5-page paper. And you're going to be reviewing about 5 recent articles. And the way I define recent is probably that all of the 5 studies were published somewhere in the 21st century, okay? So, all right? And the easier way is to pick it from an established lab because they're helping you organize the thing. And of course, when you write this paper, if it's 5 pages long and there's 5 articles, you want about a half a page to introduce the topic, and at the end, maybe a half a page to summarize the 5 findings. And then the 5 papers are in some way either singly or group summarized within those remaining 4 pages, okay? And by the way, you pick out the salient characteristics. So very often, there was a favorite comedian. He died maybe about 6 or 7 years ago. And if whatever feeling I ever felt that I was a comedian in some way, shape, or form was basically stolen from me, because this comedian took a lot of the material that I thought when I would be growing up I could say, simply because his experience. Oh, wait a second, somebody's saying, so it's comparing the 5 sources and summarizing it. Yes, so I just saw that that chat. Yes, but what are you summarizing? So let's say you're studying. It's a rodent model, and in one study they basically used 42 rats. In another study they used 38 rats. Another study they used 14 rats, et cetera, et cetera. Is that an important issue? Probably not. You know, the important thing is to differentiate how each study differed from each other in terms of what their goals were, and then what they found, and then how it sort of built on each other. So if it could very easily be that when you summarize 5 studies, it might take you 10 or 15 pages to write this. I'm telling you to distill it into a 5-page, double-spaced sort of mini review. That's what you're basically doing there, okay? And again, you can get feedback as to the topic. You can get feedback as to whether the papers that you're using are, in my estimation, appropriate or not. You can go and just go on and do that. So there's probably the easiest way to do this is, does anybody have a question? Unmute yourself and ask the question so everybody can hear that. Okay, so at this point, there doesn't appear to be questions. Oh, I see, you can't unmute. Okay. So it's going to be due either December 3rd or December 10th. I'm reading, when is it due? Okay. Let me see if there's any other chats here. There you go. So you said lesion studies don't count. That's not true. A lesion study counts if you lesion the hippocampus and then versus a control or something, and you're looking at an anatomical measure somewhere else. That counts. But if you're looking at a lesion study, let's say a lesion in the hippocampus, and you're looking at some type of behavior, that's not appropriate for this course. So we'd like to discuss the methods at length or only when they're relevant. So let's see, obviously you would, if there is all of a sudden that you found X finding over here in one paper and a totally opposite finding in the other paper, and then you found that there was some small methodological change, that becomes very important and salient. So therefore, you would talk about that. Can you give us another example of where the dependent variable is anatomical? So, so it can be, so in lots of fMRI studies or something, you're looking at what are the areas of interest that are lighting up or not lighting up, or showing increases or showing decreases. The different dependent variable can literally be looking at something like changes in cytoarchitecture. So a very classic type of change was a guy by the name of Dominic Purpura, who for a long, long time was the editor of brain research and head of the neuroscience department up at Albert Einstein College of Medicine. And what he studied was Down syndrome. And he was one of the first labs to basically show that in Down syndrome, people afflicted with Down syndrome, that one idea was, they had fewer cortical cells, in particular areas of the cortex. And they would do these very important cell counts and everything. And what would they end up finding? They ended up finding that the number of cells were pretty much the same in a Down syndrome brain than in a non-Down syndrome brain. But what differed? What differed was the dendritic branches, and they had the same number of dendrites. But what did they have? When you looked at the dendrites and you looked at the sort of tree-like arborizations and the dendritic spines and everything, that's where Down syndrome brains showed a massive reduction. And then the argument then becomes, well, was it because of the genetic aspect of Down syndrome might be changing that? And there were just, I could sit here all day and tell you about dependent variables that are anatomical. So what I would ask you to do is look at something that interests you, look up a paper, send the thing to me, and then I will, I'll then basically tell you whether you're on the right track or not. Okay? And of course, we can repeat and go back to this when I, in the coming weeks. We don't, if people want to bring this up. So now the last piece of work that we have to do before we step in, and again, I'm going to leave it to you as a group, is we have a class that we have to make up, simply because a couple of weeks ago, I had that massive power failure and I could not get to you. Another example, I see you're just telling, okay, very good. Yes, that's a good example. Yeah, always as a DV. Okay, so the other thing is picking some point, some date, and it may be almost impossible to do this, of seeing where there is a date or two dates in which, so a day, a date, and a 3-hour block of time where I can give another lecture, okay, and make up that lecture. And I say that we should do that somewhere during November, so we're not backloading all the way to the end. And that would give you a bit of time for me to catch up and for you to be able to absorb that material. I don't think we want to have that makeup class on the very, very last week, because you have other courses, you have other responsibilities, and then everything gets jammed to the end. So I'm going to ask that you discuss this among yourselves, come up with some type of a thing. And of course, what we would make absolutely sure of is that this, that the class is recorded so people can get to it. Okay? So that's the other thing you have to go and do. So now at this point, I am now going to go on and start today's lecture where we left off in the last class. So here we are in the last class. We were here and we were looking at a mid-Pontine section, and we saw all of these structures, but the two structures that I sort of zoomed in on were right along the periventricular gray of the pons. One was the dorsal tegmental area, which we can see right here, which is found very close to the MLF. That's E. And then the letter F was this small nucleus in the pons called the locus coeruleus. And the whole thing of the dorsal tegmental nucleus is it's a very good example of a nucleus which is periventricular. So what we're doing is, what I want you to think of is when you look at the ventricular system, especially starting in the third ventricle and working caudally. So we have the periventricular gray surrounding the third ventricle. We have the periaqueductal gray surrounding the cerebral aqueduct. We have the periventricular gray that we see here surrounding the fourth ventricle. And then we have the pericanular gray that surrounds the central canal of the spinal cord. When you're looking at the ventricles itself and the aqueduct, where does that come from? It comes ontogenetically, development within an organism, all the way back to very, very early development of the zygote that then forms a blastula and then forms a circle of cells that surround an opening, and within that opening you then have the development of those cells into either ectodermal tissue, mesodermal tissue, ectodermal tissue. And the point of the matter is that a lot of the neural tissue and glial tissue is coming from either endodermal or mesodermal sources. So this very interesting ventricular system, and all the way down into what we would think of an embryonic spinal cord, is very early in development. So therefore, and now if you now do that into phylogeny, and you basically now think of it moving from simple organisms to complex organisms, you can see a very simple nervous system being formed very, very early. And it is around this ventricular system. And the ventricular system, as we know, is unidirectional. It basically starts up in encephalic areas, and the CSF that's being produced is being produced rostrally, and it pushes out caudally. So therefore, not only is the CSF a nutrient, but CSF can be a way, sort of a neurohormonal way by which the rostral part of the nervous system communicates with the caudal part. So a very interesting aspect of this is that when we started to look at a class of substances, especially neuropeptides, can get released into the CSF around the periventricular area. And one of the things that I studied a lot were the opioid peptides. And not only did the opioid peptides communicate with one another through neural communication, but they also communicated through the CSF. And one beautiful example of this is that if you basically look at a female rodent, and the female rodent is now pregnant, so and she's about to give birth, what basically happens is that there's a pulsatile increase in the amount of opioid peptides that can be found in the CSF as the rodent is giving birth. And remember, with a rodent, she can give birth to 6 or 8 pups at any given time. And you will see a cyclical rise in an opioid peptide, then a fall, and all of these things are timed to the actual act of parturition. So a guy by the name of Alan Ginsler discovered that pregnant animals, as they give birth, literally will be analgesic during that time that they are expelling each and every one of the fetuses, okay? And then, very interestingly, in a related issue is, again, with a rat, every time the mother gives birth, the mother then reaches down to the pup that just was born, basically snips off the placenta, and then ingests the placenta. And what has been found in the placenta of these baby animals, an opioid-like factor that can be given to other animals that will produce analgesia that can be blocked by an opiate antagonist. So there's a very interesting hormonal process, neural process that basically uses this these areas around the periventricular, pericannular, and periaqueductal areas. And we find, just like, and we're going to revisit this when we go into the pericannular gray, that is going to be an area that's going to be very important in the mediation of stress responses. And then we're going to be looking at a number of these areas, periventricular and the Pons and whatever, that again are involved in homeostatic responses. We'll see this especially when we go into the midbrain as we talk later in this class. So now what I want to do is I want to focus on this tiny little nucleus here called the Locus coeruleus. And the reason I am basically focusing on this is, as I pointed out at the end of the last class, the Locus coeruleus is quite unique in that the cytoarchitectural structure of the Locus coeruleus, in which each cell of the Locus coeruleus is very, very similar cytoarchitecturally to its neighboring cell in the Locus coeruleus. In addition, every last one of these cells is capable of releasing the neurotransmitter norepinephrine. So every cell, cytoarchitecturally, in the Locus coeruleus has the chemical machinery by which that cell can synthesize norepinephrine. So of course, all kinds of cells get all kinds of amino acids that come into the cell. And the important amino acid that you're basically looking at for the synthesis of norepinephrine is tyrosine. And all of these Locus coeruleus cells have two very important enzymes. One enzyme is tyrosine hydroxylase, which will literally take tyrosine and with tyrosine hydroxylase produce dopamine. And then there's a second enzyme called dopamine beta hydroxylase. And dopamine beta hydroxylase changes dopamine into norepinephrine. And where is that basically happening? That is happening out in the Bhutan. So that basically what's going on here is that this small population of cells then sends out axonal projections. And what we can basically see here is we go in almost 6 different directions with noradrenergic pathways coming out of this tiny nucleus. So you can see, and we'll talk about these things later, the locus coeruleus projects up into the cerebellum. It projects down into the brainstem. It projects down into the spinal cord and innervates the dorsal horn, innervates the ventral horn, and innervates the intermediate zone, especially the area around the pericannular gray. So what we can right off see is 5 different places you can go. Then what the locus coeruleus does is it projects and forms the so-called dorsal noradrenergic pathway that as it's going forward will innervate very important parts of the midbrain, including the Raphe nuclei and the dopamine rich nuclei in the ventral tegmental area and in the substantia nigra. It then reaches up into the hypothalamus and it innervates the paraventricular nucleus. And then it goes up into the limbic areas like the amygdala, the septum, and then it goes all the way up into the hippocampus. And additionally, this pathway ends up going to all 4 lobes of the cortex. So there's just an enormous array of places. So where the locus coeruleus starts to release norepinephrine, it is literally like a little alarm clock. Now, what's very interesting is the way it does this is that in many of the cases, when you're looking at it electrophysiologically, is that the norepinephrine, when it's acting on a cell and a receptor, very often it will produce inhibition. But what is it basically doing? It's inhibiting a cell that in turn inhibits something else. So the process that you're getting, even though there's electrophysiological inhibition, it is functional disinhibition. So all of a sudden, you very often see the locus coeruleus and norepinephrine involved in arousal, attention, things like that, a shifting towards a salient type of stimulus. So very often you'll see the locus coeruleus involved in particular mediations of learning. And the biggest area where you basically see this is that the locus coeruleus projects up to the hippocampus. And how it acts in the hippocampus is it acts to modulate a particular type of general hippocampal activity, which is called hippocampal theta. And what has been found is that hippocampal theta very often is associated with the memory consolidating properties of the hippocampus, okay? So in a lot of ways here, we see that, like I call it, the little engine that could, this very small nucleus has sort of an outsized effect. Now, the way it does that is another interesting property. Very often, what we think of when we think of an axon coming out of a cell body, we basically think of the axon going to some endpoint and then releasing whatever it's releasing. a corollary of that, sort of what you can see in this diagram here, is you can basically see an axon that goes along, but then has a whole bunch of sort of collaterals that go off. So if you have the main axon here, it might go to 5 different places and then release norepinephrine. That's another way. But a third way that's very interesting is that the locus coeruleus fibers are what they basically call a leaky hose. So if you imagine that somebody has cleats on, those golf cleats that have little points on them, and you walked and you stepped across a garden hose back and forth, and now turn the garden hose on, not only does the water come out of the end of the garden hose, but they sprout things along the way. So what the locus coeruleus can be doing is releasing norepinephrine not only at its endpoint terminal, but it might be releasing norepinephrine along the way with all of these very relevant, important structures that it passes. So in a lot of ways, you now get into a sort of another very, very vague term, which is called neuromodulation. So when the locus coeruleus cell fires and it releases norepinephrine at its terminal, yes, that's a punctate effect, but it can also be releasing norepinephrine all along the way that then modulates the activity of all of those things along the way, along with its endpoint. So that is a norepinephrine pathway to locus coeruleus. We'll come back to this later. Another example is that norepinephrine is not only synthesized in the locus coeruleus, it is synthesized in a group of other medullary and pontine nuclei. And 2 researchers by the name of Annika Dahlstrom and Kjell Fuchse, almost 55 years ago, did the seminal study in which they basically described the noradrenergic cell structure. It wasn't all over the place. It projects all over the place, but they're punctate nuclei. And what they ended up doing is identifying what they call A group cells, which is containing catecholamines, norepinephrine, and dopamine, B type cells that contain and release serotonin, and C type cells that release epinephrine. And what they basically found is rather than these cells being ubiquitous throughout the brain, they were localized in these very important areas. So the locus coeruleus is called A6. So that means there's an A1, A2, A3, A4, A5, and A7. There's 7 noradrenergic nuclei. One of them is locus coeruleus. The other 6 are found in the ventral part of the Pons, all the way back to the medulla, in this heretofore unknown area called the nucleus reticula, or the reticular formation. And what you basically find is, although the locus coeruleus has like 6 or 7 different, depending on how you argue it, terminal areas, the ventral noradrenergic cells, even though they're distributed in different parts of the medulla, like around the nucleus tractus solitarius, all the way up to the rostral pons, they form what is called the ventral noradrenergic pathway. And that basically goes more into the lateral part of the hypothalamus, goes into the limbic system, and then goes up into the 4 different parts of the cortex. So if you remember the reticular formation, where I basically talked about something having to do with consciousness and awareness and orienting and all of that, you can see the important role of these ventral noradrenergic pathways. But the cells that just synthesize norepinephrine simply because they take tyrosine and use the enzymes tyrosine hydroxylase and dopamine beta hydroxylase to produce norepinephrine, those cells are found in the medulla and the pons. Let me, okay, what I didn't include here is there are now cells in the midbrain and diencephalon that are catecholinergic. And I'll cover that a little later when I talk about the midbrain. And what you're going to basically see here is that these cells, again, can synthesize tyrosine, but they have only one rate-limiting enzyme, the tyrosine hydroxylase, that allows for the production of dopamine, and they get released. And we'll talk about them when we get into the midbrain. But again, now we can look at the serotonergic cells, and we started to get introduced to this when we basically looked at the Raphe nuclei that are found right along the midline, as far caudal is the caudal medulla, and they go as far rostral as the midbrain. And they synthesize the neurotransmitter serotonin. And the way they basically do that is they take the amino acid tryptophan and enzymatically change it, mostly through tryptophan hydroxylase to form serotonin. And as we pointed out, we are going to have roughly in a rat, we have 9 different Raphe nuclei. The most caudal of them mostly descend down into the spinal cord. The middle 3 very often just innervate the cerebellum and the surrounding medulla and pons and midbrain. And then the rostral 3 are going to ascend and innervate things like the thalamus, the limbic system, and primarily the frontal cortex of the brain. And again, we'll be covering this. So now, while we're on the subject of neurochemical neuroanatomy, there was another major neurotransmitter that started to become identified. Early in the process, people started to look at the cholinergic system on the basis of whether or not a particular brain area held the enzyme that metabolized acetylcholine. And that enzyme is called acetylcholine esterase. And basically what able to find is that acetylcholine esterase was found in many parts of the brain. So it was thought, just like the excitatory and inhibitory amino acids, acetylcholine must have been everywhere. But we did not have, with histofluorescence, the ability to look at acetylcholine because the histofluorescence technique did not pick up things like these rate-limiting enzymes. The major rate-limiting enzyme that takes the amino acid choline and creates, eventually creates acetylcholine is choline acetyltransferase. C, big C, little h, capital A, small t, chat. The way this was found was when finally we're able to look at immunocytochemistry, which I talked about briefly before. And that is where you could take a substance and then create an antibody to that substance. And then by using that antibody label, you could then begin to identify what cells in the brain contained that rate-limiting enzyme, CHAT. And what was then found is that the idea that acetylcholine was ubiquitous because of acetylcholine esterase was dispelled. And in fact, what you basically find is that there are really only two major cell groups for acetylcholine. One is right around the area of the Pons and the midbrain. And this is given the wonderful brief name of the laterodorsal and pedunculo-pontine tegmental nuclei. Let's take that apart. Tegmental means floor. Pontine, it could be the pontine area, and pedunculo. We're going to see in the caudal midbrain a structure called the interpeduncular nucleus. So the cholinergic cells are found in the caudal midbrain and pons, so pedunculo-pontine, and the floor. And then there's also a lateral dorsal division. So what you can basically see is that this group of cells are sending acetylcholine fibers to the cerebellum, to the superior and inferior colliculus, to a structure called the medial habenula, the thalamus, the dopamine substantia nigra, to the whole pontine reticular formation, to the locus coeruleus, the vestibular nuclei, to the Raphe nuclei, and then down into the medullary reticular formation. So there's this very interesting thing. And one thing you start to notice here that you probably learned in any kind of behavioral pharmacology course is that some of the major neurotransmitters interact with one another. That is, norepinephrine interacts with dopamine, norepinephrine interacts with serotonin, acetylcholine interacts with norepinephrine, acetylcholine interacts with serotonin. And here, what you begin to see is how this nature of this interaction happens anatomically. But the very interesting group of cholinergic cells are basically found very rostrally in an area called the nucleus basalis, the medial septum, and then the basal ganglia, both the dorsal striatum and the ventral striatum. And what this actually is a big long band of cholinergic neurons. Now this becomes very important because of where these projections end up. As you can see, the dorsal and ventral striatal cholinergic areas are primarily interneurons, whereas the medial septum and the nucleus basalis project to all four parts of the neocortex. They project to the limbic system, and very importantly, they project to the hippocampus. And of course, it was this discovery right around the late 1970s that then was paired with whole bunches of psychiatric studies and basic neuroscience studies that implicated acetylcholine in the modulation of a very important debilitating disease. And that disease is Alzheimer's disease. So what you can basically see is that from the medial septum, the cholinergic cells will project up into the hippocampus that then start to interfere with the consolidation of short-term memory. And the whole thing having a lot to do with the type of confusion and things like that that you see in Alzheimer's disease. And then what you basically see is this connection up to the cingulate cortex and the neocortex. And of course, this is not the only story, but the cholinergic theory of Alzheimer's disease became one of the first major players, just like you have a serotonin theory of depression, you have a dopamine theory of schizophrenia, et cetera, et cetera. But so what you have here is not only the emerging neurochemistry, but the emerging neurochemistry that you then pair with the underlying neuroanatomy. Okay, so here are various types of techniques that you basically have, and you can look at them in your textbook. You can look at them, Google them and everything. By the way, you can use these techniques very, very much in any one of your papers. So histofluorescence, immunocytochemistry, receptor orthoradiography, where you're identifying where receptors are. Then the more advanced techniques of in situ hybridization. Then what you can do is use proteins as a marker for both orthograde and retrograde tracing techniques of one site impinging on another, and then a more advanced thing like mRNA as a marker that can then look at that. And of course, there's a whole host of newer types of techniques that now look at transneuronal connections with a high degree of selectivity. So now we bring ourselves to the rostral pons. And one of the things that you're starting to look at right now is something different. So first of all, let's again look at the rostral pons as to what's there and what's not there. So one of the things is we basically are starting to look at the shape of the ventricle. That as we looked, when we finally emerged out of the spinal cord and the medullary spinal transition, we saw the central canal moving superiorly and the dorsomedial septum opening up like a chevron, so that you formed a horizontally aligned fourth ventricle that basically was the roof of above the medulla and the pons. Now, when we get into the rostral pons, what are we now starting to see? We're seeing this horizontally aligned fourth ventricle actually starting to start to look more like a hole, which of course, as we move rostrally, will be the cerebral aqueduct. Also, what we now see is that the fourth ventricle no longer forms, the roof of the fourth ventricle is no longer the vermis of the cerebellum, but rather we're starting to see these two protuberances, which of course we know in the midbrain as being the inferior colliculus. And we see that there is actually neural tissue, tectal tissue above the fourth ventricle here in this rostral pontine section. Then what we basically see in here is, again, of course, we have where's my pointer? Yeah, come on, here we go. The periventricular zone, all of those nuclei we talked about before. We still have the MLF, we have the Raphe, and we have the horizontally aligned medial lemniscus. So what's above this is the reticular formation. What is below this is the Grisium pontus. We still have the middle cerebellar peduncle at the bottom, and we have the corticospinal tract. And then what else do we have here? What we have in this rostral pontine section, of course, is the trigeminal nerve, which is the fifth cranial nerve, which is mixed. the sensory portion is lateral, the motor portion is medial. So here in this rostral pontine section that we actually look at, we can basically see some of these important structures. And in this rostral pontine section, we're actually now going to start to see some midbrain type of things like the trochlear nucleus. And of course, we have the trigeminal, etc. So here we go. So we have the cerebellovermis. We have the dentate nucleus of the cerebellum. We have the superior cerebellopeduncle here. Remember, that's coming back from the midbrain after it decussated and is going into the cerebellum. We have R, which is the middle cerebellopeduncle. And then we have the MLF. And we actually have right here the trochlear nucleus. We then have what we would call either the fourth ventricle or the beginning of the caudal cerebral aqueduct. Then H, we have the trigeminal nuclei. O, we have the reticular formation. L, we have the medial lemniscus. Q, we have the Grisium pontus, and embedded within it is P, which of course is the corticospinal tract. So now when we go into this ponto midbrain transition where we move even a little bit more rostrally, now you're really starting to look like a midbrain, but not quite, okay? That's why we call it a transition. So first of all, what we see the big difference is, look at the size in this rostral pons of the fourth ventricle. Now look here at B, and what has happened, the ventricular system looks more like an aqueduct. You can see a much larger superior cerebellopeduncle. What else do we see here? Well, something that is very interesting here. K, L, M, and N is the medial lemniscus. Notice how the medial lemniscus is moving more laterally. But what is I? I is the lateral lemniscus. It was lateral in more caudal sections. Okay, let me get back to my. I, in more caudal sections, was sitting lateral to the lateral lemniscus. But what is I doing, the lateral lemniscus? What it is basically doing here is that it is, why am I having trouble here with my pointer? With I, you're having the lateral lemniscus eventually moving up to where? The inferior colliculus, where it's going to synapse. So remember, the lateral lemniscus has all of those auditory inputs that originated in the dorsal cochlear nucleus, ventral cochlear nucleus, superior olivary nucleus, and trapezoid nuclei, moving up and going into the lateral lemniscus. But is this a true midbrain section? The answer is no, because we don't have a cerebral peduncle yet. We still have remnants of the middle cerebellopeduncle here, and we can see the corticospinal tract embedded within the Grisium pontus. So there we are. That is the midbrain, or the pons. So at this point, Let me look at this for a second. All right, we're at 10:30. Why don't you take a little break? And then when I come back, we're going to enter into the midbrain. Okay. Okay, I am back. Let me just check if I'm assuming that everybody is back, so we can start again. So now share my screen. Okay, so we're going to now enter into the midbrain. Now, one of the things that if you just think about 4 different areas, the midbrain, the pons, the medulla, and the spinal cord, the very interesting thing that the midbrain and the spinal cord share is the relationship of it to its relative thing of the ventricular system. So if you remember in the spinal cord, what we were really able to do, because the central canal was right at the center, we were able to identify things that were sort of dorsal to the central canal. So the dorsal horn and things and the various structures there. Then we were able to identify things that were ventral to the central canal, the ventral horn, etc. And then we had an intermediate zone. In the pons and medulla, we did not have that ability simply because the 4th ventricle really separates the cerebellum from the underlying pons and medulla. So everything in pons and medulla is by definition tegmentum. But in the midbrain, again, with the cerebral aqueduct, the cerebral aqueduct doesn't sit in the dead center of the midbrain, but at least it sits in the midbrain. So what we can see is structures that are above it that we literally call tectum or roof, and structures that are below it, which we can call tegmentum. The other thing that we can see is with the central canal, we had the pericannular gray completely surround that central canal. And like I said, in future classes, when I start talking about some homeostatic systems, we're going to be going back to that central canal and talking about that. But very much here, in where we have the cerebral aqueduct, we then have what is called the periaqueductal gray that literally looks very much like a keyhole that we're basically going to see. Okay, oh, I see, I did something wrong there. Let me get out of this for a second, then go back. Now come here, there we go. So here, one of the first things, because we're in the caudal midbrain, and we started to see this even in that transition area, is the inferior colliculi. And of course, that's found on the superior part of the caudal midbrain, here and here. And that the inferior colliculus is an important part of the auditory pathway. So as we pointed out, the auditory pathway is entering in through the cochlear branch of the vestibular cochlear nerve, where the inner ear and the cochlea is putting its synapses onto the cochlear nucleus. From the cochlear nucleus, what we're able to have is outputs that go to the ventral cochlear nuclei, the superior olivary nuclei, the trapezoid nuclei, and eventually what you then form is a common pathway called the lateral lemniscus. And the lateral lemniscus, as we saw, has both ipsilateral ear input and contralateral ear input. But the contralateral ear input is roughly about 60%, and the ipsilateral ear input is roughly about 40%. And what these lateral lemniscal fibers do, they are basically out here and here. They're coming in and synapsing in the inferior colliculus. So again, as I pointed out, if we were amphibians or whatever, this would be the end of our auditory pathway, and that would be that. But of course, we're not amphibians, and what we have done is developed two more steps in the auditory pathway. And the first of these steps is the medial geniculate body of the thalamus. And the way that fibers go from the inferior colliculus to the medial geniculate body of the thalamus is through what is called the brachium of the inferior colliculus. So we've seen this word brachium, like brachium pontis and things like that, and brachium conjunctivum. We're basically talking about fibers here. And then what we're going to see, and we'll see this much more when I talk about the thalamic nuclei, the medial geniculate body then will send its projections through the temporal radiations to the superior temporal gyrus. Okay, so while we're sticking with the tectum, let's now move to a more rostral structure, and that rostral structure is the superior colliculus. the more rostral of the two protuberances of the corpus quadrigemini. Now, the superior colliculus has an enormous amount of other systems going on. Sensory motor integration can happen in the superior colliculus. But a very important part of the superior colliculus has to do with vision. So what we basically saw early on when we talked about the cranial nerves is that what we basically saw is that the cranial nerve, the second cranial nerve, when you have the optic chiasm in the human being, 50% of those medial fibers of the optic nerve literally go and cross and decussate at the optic chiasm. The other 50% that are more laterally organized, those ganglionic retinofugal fibers, basically stay ipsilateral. Now, after we have the optic chiasm, these ganglionic retinofugal fibers can go one of three different places. The vast majority, well over 90% of these ganglionic retinofugal fibers will go to the lateral geniculate body of the thalamus. And then eventually the inputs that go into the lateral geniculate body of the thalamus will then form axons that go through Meyer's loop and the occipital radiations back to the primary visual cortex. We'll be talking about that, especially when we talk about the thalamus. But then about 5% of the fibers, 5 to 7% of the fibers, literally these ganglionic retinofugal fibers go into the superior colliculus. Then about 2% of the fibers, ganglionic retinofugal fibers, go into the suprachiasmatic nucleus of the hypothalamus. Look at the name of that. It basically tells you where you are. It's a nucleus, and supra means above, and it's above what? The optic chiasm. So a very small number of these fibers that are decussating in the optic chiasm literally will go to the suprachiasmatic nucleus. And what is the suprachiasmatic nucleus important in? So let's go and basically think about an organism. An organism eats. An organism drinks. An organism is active. An organism is inactive. An organism has sexual and aggressive behaviors and whatever. The suprachiasmatic nucleus, if you lesioned the suprachiasmatic nucleus, you would not change the amount of any of those behaviors. You would eat the same, you would drink the same, you would have sex the same, you would have aggression the same, you would have pretty much the same amount of activity and inactivity. But that tiny little nucleus basically determines when you have that. So what the suprachiasmatic nucleus basically does is it's involved in circadian rhythms. And we'll talk about this later when we go into the hypothalamus and when we talk about probably the prototypical animal to study this activity. Whereas we saw with consciousness, people like to study cats. With feeding, people like to study rats and mice. But in the suprachiasmatic and circadian rhythm, the creature people like to study is the hamster. And we'll basically go and look at that. So the superior colliculus is basically getting about 5% of the input. Now, what becomes very, very important here is that what happens if all of a sudden the lateral geniculate body of the thalamus bilaterally stops working, or that you have a big stroke that basically affects the primary visual cortex and basically destroys it. You've seen things like the calcarine artery. Let's say something like that happens. There's a devastation there, and all of a sudden, the primary visual cortex just basically dies. So the question is, are you blind? No, you're not, because you still have the suprachiasmatic nucleus, and very importantly, you still have the superior colliculus. But what you basically have is an achromatic type of world. You see things in black and white, but mostly gray. And then all of the very important types of things that needs binocular information, like the like depth perception and things like that, goes away. So you have an achromatic flat world, but you can see movement and everything like that. And why? Because the superior colliculi in the amphibian brain is what? The endpoint of an amphibian visual system. Okay, so that is the superior colliculi. So now let's look at a caudal midbrain slide. We're going to be looking at the inferior colliculus and lateral lemniscus. We're going to be looking at the trochlear nucleus, the MLF, the Raphe. Then we're going to start to see the superior cerebellar peduncle, the medial lemniscus and what its shape is, and the Grisium pontus. So here we go. Here is a schematic. So first of all, what we basically are now starting to see is some aspects of what you basically learned about in gross anatomy about a midbrain, but then some that you didn't. On the superior surface, we see the inferior colliculus, and what we see coming in here is the lateral lemniscus synapsing on the cells of the inferior colliculus, and then their outputs giving rise to the brachium, into the brachium of the inferior colliculus. We then have the periaqueductal gray, sort of looking like a keyhole, and a very clear cerebral aqueduct. So what we're going to see is a dorsal periaqueductal gray, a ventrolateral periaqueductal gray, and then we're going to see a ventral periaqueductal gray. Well, right in here, what we can see in the caudal portion of the midbrain is we have the nucleus of the fourth cranial nerve, the trochlea. And what we have found is that the trochlea is the second of three cranial nerve nuclei that innervate eye muscles. It specifically innervates the superior oblique eye muscle. And the other point about it is its bizarre trajectory. Whereas all of the other 11 cranial nerves either exit the brain laterally or medially, they're leaving the brain in a ventral direction. What does the trochlea do? It exits the brain dorsally and medially. So it comes up to here around the superior colliculus, and then it will shoot out laterally. In that space between what? Where the sort of cortex ends and the cerebellum begins. That's where you can find that trochlear nucleus. And of course, what's very interesting is if you basically, if you imagine a trochlear nucleus as a pair of eyeballs, you have little bags under your eyes right here. And what is that? That's our good old friend, the fiber tract, the MLF. Remember, the MLF is coming from the medial vestibular nucleus and has bifurcating axons that go all the way back into the medulla and all the way up into the rostral midbrain that is giving you positional information. So this positional information becomes very important with the trochlea in terms of whether or not that trochlea muscle should be engaged, dependent on whether your head is straight up or it's to the side or it's at some angle. So then what we again have is the dorsal PAG, lateral, ventrolateral, and then the ventral PAG. We now start to see some of the Raphe nuclei here. So one big one that we're going to see here is the dorsal Raphe nucleus, and then we're going to see the Raphe linearis. Now, the dorsal Raphe nucleus has two types of projections. In one type of projection, the dorsal Raphe nucleus will send descending projections, and it will innervate another medullary Raphe nucleus called the Raphe magnus. And the dorsal Raphe nucleus and the nucleus Raphe magnus are the major sources of serotonin control over pain inhibition, especially that type of pain inhibition that reaches down into the dorsal horn and blocks the sensory discriminative aspects of pain. By doing what? Sending these projections down into lamina 2 and lamina 5 of the dorsal horn and synapsing presynaptically on nociceptive fibers coming in. So basically what it's doing is serotonin, by inhibiting these fibers, will literally stop them. Now, the dorsal Raphe nucleus also sends fibers, serotonergic fibers, into the nucleus Raphe magnus. And what did I talk to you about with the nucleus Raphe magnus and these medullary Raphe? That they have three different types of cells having to do with pain and pain inhibition. On cells that inhibit pain, off cells, on cells that activate pain, off cells that inhibit pain. And how? Because there's an interaction between serotonin and endogenous opioids in inhibiting, and then neutral cells. And then in addition, the dorsal Raphe nucleus is going to ascend and go primarily into the limbic system. Then what we have below here is the Raphe linearis. And the Raphe linearis is interesting, again, being serotonergic, and in all of these cases, roughly only about 1/3 of the cells are serotonergic. But what the Raphe linearis does is project rostrally into the limbic system and into the frontal cortex. And there's another interesting aspect about the Raphe and serotonin that is involved in a very powerful developmental disorder. And that powerful developmental disorder, of course, is ADHD and ADD. So ADHD is attention deficit hyperactivity disorder. ADD is attention deficit disorder without the activity. And there are many models of this that are exquisitely sensitive to serotonergic function and into the Raphe linearis. So with the Raphe linearis, if you go and you destroy that, you can end up with a permanently hyperactive animal. Okay, so that is the whole periaqueductal gray. Then what we have are what looks like two different balls sort of pulling towards one another. That is a sort of schematic of the decussation of the superior cerebellar peduncle. Now, of course, surrounding the periaqueductal gray and the superior cerebellar peduncle is, again, you have midbrain reticular formation. And now what you have is at the base of the reticular formation is the medial lemniscus. And now look at that medial lemniscus. It's now more and more lateral. And instead of being horizontal, it is now sort of forming into a boomerang. So it becomes a very distinct type of structure that you're able to basically see through the midbrain and sort of give you an idea as to where you are. Then below it is you still have some greasy and pontus, and you have the corticospinal tract. So even though we call this a midbrain section, on its ventral surface, we still see some brachium pontus. We're not seeing cerebral peduncle yet, okay? So now we basically move to a caudal midbrain site. And again, we see a lot of these structures, and here we go. So what is A? A is the inferior colliculus. What is C? Is the brachium of the inferior colliculus. What is D? And seeing over here is the lateral lemniscus. So the lateral lemniscus is carrying auditory input where its synapses on A, the inferior colliculus, and then the axons of the inferior colliculus then form the brachium of the inferior colliculus, and they are eventually going to go up to where? The medial geniculate body. Then what we see is a very, very clear K, the cerebral aqueduct, and what we can see is a very clear periaqueductal gray done by the letter L. And you can start to see the sort of keyhole here, which is the sort of Raphe nuclei. Then what we have, just in the ventrolateral portion of the periaqueductal gray in the caudal midbrain, is the letter M, which is the trochlea nucleus. And of course, just adjacent to the trochlea nucleus is the fiber tract, the medial longitudinal fasciculus, which of course is modulating the activity of the trochlea nucleus. Then we can look at here, M, what we're basically seeing right here is the decussation of the superior cerebellar peduncle. Out on the edges here, we're seeing parts of the midbrain reticular formation, but you can see the decussation of the superior cerebellar peduncle as very, very important. The descending fibers are going to go back down into the cerebellum. The ascending fibers of the superior cerebellar peduncle are going up into the thalamus, up into the cortex, etc. Now, then what we see, let's focus on G, E, F, and G. What we see right here now is our medial lemniscus. Here is the medial lemniscus right here. Here is the medial lemniscus. And now, finally, what we're able to see on the base of the brain, labeled as Q over here, and then labeled as STUVW, this is just a label showing that it is somatotopic, is, of course, the cerebral peduncle. So what is this? This is the corticospinal tract as it is coursing through the midbrain after the crus cerebri on its way to the corticospinal tract in the pons. Then it will appear on the inferior surface in the brain in the medulla. And then in the caudal medulla, you will have a 90 to 95% decussation to form the lateral corticospinal tract and the anterior corticospinal tract. The other thing we have here in the very caudal midbrain, marked by the letter X, is the interpeduncular nuclei. And remember, I mentioned with the cholinergic system, the pedunculopontine cholinergic nuclei. Now, again, it's very important for us always to remember that what are we looking at here? What we're looking at is a stain, which is a Wheal-Weigert stain. And a Wheal-Weigert stain, of course, stains myelin. And it stains the myelin, which is typically white matter, dark, so it makes the gray matter look lighter, okay? So if there's ever a thing where you basically are saying, well, the white matter is dark and the gray matter is light or white, here we have probably the strangest way of taking a name of a structure and in showing it on a Wheal-Weigert stain, it's sort of the opposite. What is the letter P? The letter P is a very large nucleus or nuclei in the midbrain, going from the caudal midbrain to the rostral midbrain. And it is called the substantia nigra, the black substance. Yet we look at it in a Wheal-Weigert stain and it looks white. If you didn't have it stained at all, what you would basically see is that nigral tissue sort of has this slight pigment towards a darker kind of color, hence called the substantia nigra. And of course, the substantia nigra, together with another nucleus that we're going to see very soon in the same trajectory of where X is, the interpeduncular nucleus, is called the ventral tegmental nucleus. And the substantia nigra and the ventral tegmental nuclei contain the neurotransmitter dopamine. And what you basically are going to see here is two major aspects of what we consider an important modulatory role for dopamine. The one area in which we consider a modulatory role for dopamine, of course, has to do with movement. And that is primarily an area having to do with the substantia nigra. And then a second modulatory role for dopamine is in the whole area of motivation, okay? And very interestingly, a famous neuroanatomist asked, when looking at the whole thing of these dopamine cells and where they project, and then what happens, because we're going to be talking about a lot of this when we talk about the limbic system, when we talk about the striatum, and when we talk about the cortex and all of their myriad interactions. The neuroanatomist Wally Nauta, N-A-U-T-A, basically asked the simple question: where does motivation begin and where does motivation end and motor begin? And this, of course, always was an interesting question to me. And as I was doing a postdoc up at Columbia, just before I got the job at Queens and started teaching neuroanatomy here, and always being interested in motivation, I was a record album came out, and the record album was written by the whole album. Was written by a guy named Jim Steinman, and Jim Steinman looked around and tried to find the right vehicle by which he could take this record album and put it out. And he found the perfect person. Again, I'm not going to ask you to raise your hand or anything, but how many of you have seen the movie that was created in 1974 called The Rocky Horror Picture Show? Okay, so if you've never seen the movie, The Rocky Horror Picture Show, you can go to Google, go to Wikipedia, and basically look at it. It's an utterly bizarre movie that starred a young Susan Sarandon and a couple of other folks, and it introduced the character Tim Curry, who played Dr. Frankfurter, and you'll figure out the phallic reasons for all of that later. And what ended up is it went out, and then it became one of the first cult movies of all time. So that for something, and it may still happen, although in the age of the pandemic, it doesn't happen, that on any given Saturday night in a cool part of any kind of city, people will flock to the movie theater to watch the showing of the Rocky Horror Picture Show and basically recite all of its lines. And one of the characters that came out of that was this motorcyclist named Eddie. And he was played by an actor that went along with one word. His name was Meatloaf. And you can watch Eddie in the Rocky Horror Picture Show and the wild, uninhibited kind of singing he does. So basically, you now have Steinman with this record album that he wants to put out. And that record album was called Bat Out of Hell. Again, if you've never heard of it, go look for it and whatever. All of the stories, all of the songs are basically stories that are stories of teenage angst. The most famous one is Paradise by the Dashboard Light. I encourage you to listen to it, to watch it, to read its lyrics. But right here, where I basically talk about brain dopamine, and we talk about its powerful modulatory role in motor and its powerful modulatory role in motivation, and then we get up to the point of now this question, where does motivation end and motive begin? Lyrically, one of the greatest songs that basically asks this question in rock history was the song All Revved Up with No Place to Go. And it's basically about a 14, 15, 16-year-old boy who can't drive yet, can't go out yet, and you just have all of the libido and all of the built up, when you just listen to the song and you do it, you basically can go and look at this neural circuitry and see, I have to get going and then burst out and explode and whatever. So anyway, this is how my bizarre brain works and everything like that in terms of tying things that are anatomical, things are behavioral, and what is basically going on here. Okay, so now we move to the middle of the midbrain. So in the what we learned when you went through the cranial nerves is you saw that the fifth cranial nerve was in the pons. We saw the fourth cranial nerve in the caudal midbrain exiting dorsally. And now when we get to the fifth cranial nerve, at the third cranial nerve, we're now in the middle of the midbrain. So what we're going to be looking at is for what we call the oculomotor complex, which is made up of, as I mentioned before, the Edinger-Westphal nuclei and the oculomotor nuclei. So what we're basically going to see is the third cranial nerve, and then very clearly, what we're also going to be able to see is how that third cranial nerve, both the Edinger-Westphal component of it and the oculomotor nuclei component, course out and course medially and ventrally to form the oculomotor radiations. So remember that the Edinger-Westphal nucleus is basically controlling the iris muscle, and whether the iris muscle is constricted so that the circular muscle pulls away to open up the opening, which is pupillary dilation, or whether the iris muscle is completely relaxed, in which now the muscle takes up that opening and is a smaller opening, and we have pupillary constriction and less light entering. And then we know about the oculomotor nuclei having 4 of the 6 eye muscles, the medial rectus, the superior rectus, the inferior rectus, and the inferior oblique muscle. Plus it controls the eyelid. So in an enormous amount of eye movement control and the size of the pupil in terms of letting light in is being controlled. And then, of course, what we're going to also then start to see is the close apposition of the MLF, the medial longitudinal fasciculus, that is giving us information about the position of the body, position of the head. Then what we're going to see here is another very major pigmented structure, the red nucleus. So what I pointed out to you when we were talking about control of muscles is that when we look at the descending control of muscles, it comes from 3 different pathways. One is the reticulospinal pathway. The second is called the rubrospinal pathway. And rubro means red. So where is the cells of origin of the rubrospinal tract? The red nucleus that we find in the midbrain. And then finally, the ones we've been talking about all the way along is the corticospinal pathway. And again, to differentiate these things in terms of movement, we can basically see that reticulospinal movement really talks about the proximal portion of our limbs. So it's basically controlling the shoulders and the arms. So what it basically deals with is reaching, okay? Whereas the rubrospinal tract is more distally controlled, therefore controlling the fingers and whatever, and has less to do with reaching where we go than how finally we're able to do something when we get there in terms of palpating or picking up something. And then, of course, the corticospinal tract, and the importance of it is basically modulating, clearly modulating the reticulospinal tract through the corticobulbar system, the rubrospinal tract through the corticobulbar system, and then modulating things directly through the lateral and anterior corticospinal tracts, so that you now integrate how far you're going with the very fine movements of what you're doing when you get there, okay? So everything sort of ties in together there with the red nucleus, the reticulospinal tract, and the corticospinal tract. And then in this midbrain slide, we're going to see the ventral tegmental area and nucleus and the substantia nigra. And then the last thing we're going to see is the cerebral peduncle. Of course, we're going to see things like the medial, the medial lemniscus and everything. So as you know, I have famous sort of responses to particular illustrations where I have basically seen in the past ducks looking away from each other. We've seen Christmas stockings and things like this. I call this one my Mickey Mouse slide. So, and the Mickey Mouse slide is going to be very interesting because we're not going to talk very much about the head as the Mickey Mouse slide as the ears. So what is this? First of all, when we're looking at this middle midbrain, now we're seeing more superior colliculus, okay? So where are these inputs going to be coming from? Superior colliculus that has to do with movement. It's going to be coming out of the basal ganglia, which is rostral to us. And the visual input, where is it coming? It's coming from the optic nerve and everything, which is rostral to us, and coming into the superior colliculus. And what the superior colliculus also has within it is interesting interhemispheric communication. So when we did gross anatomy, we basically talked about three major sources of interhemispheric communication. That is one side of the brain communicating with the corresponding other side of the brain, not so much rostral caudally, but going across. And of course, the most major of the interhemispheric communicators is what? The corpus callosum. And of course, we're going to spend a lot of time on that as we go on later. But then we had two other forms of interhemispheric communication. The anterior commissure, which is quite rostral to us, and now the posterior commissure. And the posterior commissure can be sort of shown right here that from the middle of the midbrain to the rostral midbrain, this is where there is interhemispheric communication. Okay, so this is not only the superior colliculus, but we also have the posterior commissure. Then what we see is a very large and clear periaqueductal gray with the cerebral aqueduct. And then what we see right in the ventral portion of the middle of the midbrain is the oculomotor complex. The most medial of this nucleus is the Edinger-Westphal nucleus, which is basically controlling the motor control of whether the iris muscles are relaxed or that they're constricted. And of course, what's very important with the iris muscle, and by the way, you start to see this in some bizarre types of drug-induced situations, is where all of a sudden one eye is differentially dilated from the other. What the hell's going on here? What's happening is some major deficit, either temporary or permanent, of the Edinger-Westphal nucleus, which of course is going to totally screw up any type of binocular vision and have the effect of differential input of light and retinal input into each of the eyes. So that's the Edinger-Westphal. And then, of course, in the ventrolateral portion, what do we have here? We have the oculomotor nerve. And the oculomotor nerve, again, is controlling those four major eye muscles plus the eyelid. And it's doing it in what kind of manner? A conjugate movement, so that whenever some subsets of those eye muscles are being altered in one eye, they are conjugately altered in another eye. If you don't have it, all of a sudden your two eyes are going either in different directions, or one eye's movement and the other is not, et cetera, et cetera. So you need not only the fact of the movement of the muscles, but the movement of all of these muscles in a conjugate, synchronous kind of manner. And then what you end up with is the sort of whiskers of of the sort of Mickey Mouse slide, which of course is the optic radiations, oculomotor radiations. I have to correct myself. So this is the oculomotor radiations that become very clear. Then what we see, and of course you see the MLF very close to the oculomotor complex. Then what we see again is the Raphe linearis, which of course again is ascending up into serotonergic input into the limbic system in the cortex and modulating in some major way activity. And then I basically see myself in a bowling alley here. What I see right here is the so-called bowling ball that we're looking at right here is the red nucleus. Look at its size, it's enormous. And then what we have is a sort of weird looking bowling pin right here. And of course that bowling pin-like structure that has still a boomerang shape and is moving out more and more laterally is the medial lemniscus. Why is the medial lemniscus moving into that shape? Because that's going to assume the shape of the BPL thalamus. Remember, the medial lemniscus are the fibers of the nucleus gracilis and nucleus cuneatus, as well as the trigeminothalamic fibers moving up and coming into the BPL and BPM thalamus for somatotopic communication into the second synapse, the thalamus. Then what we have here is we see that medial lemniscus, and then we see on the inferior surface, the inferior surface, the cerebral peduncle. But between the medial lemniscus fiber tract and ascending fiber tract and the cerebral peduncle, a descending fiber tract, we have this enormous structure here of cells called the substantia nigra. Now, if you notice, the substantia nigra at this point is now divided into two different parts. This more dorsal part is called the substantia nigra pars compacta. And it's called that because most of the cytoarchitecture of the pars compacta is largely cells. And about 50% of those cells are going to contain the neurotransmitter dopamine. So there's a dopamine projection and a non-dopamine projection of the substantia nigra. The dopamine projection of the substantia nigra coming out of those pars compacta cells form what is called the nigro-striatal tract. Nigro-striatal tract. So what does that mean? The fibers are starting in the substantia nigra pars compacta. They are ending up in the striatum. And specifically, they are ending up in what is called the neostriatum. Neo meaning new. And that encompasses two very large nuclei of the basal ganglia, the caudate nucleus and the putamen. So these nigrostriatal tracts innervate the caudate and the putamen. And what they are doing is the largest number of fibers in the nigrostriatal tract are dopaminergic. And they are projecting up into the caudate and the putamen. And dopamine is getting released there. And what we're going to find out is when we go into the basal ganglia and look back the other way, we're going to be basically finding that there are two different types of dopamine receptors that receive this incoming dopamine, a D1 receptor and a D2 receptor. And they then get divided into subfamilies and whatever. And therefore, how dopamine is going to affect different parts of the neostriatum is going to depend upon whether the dopamine is binding to a D1 receptor or a D2 receptor. And the big difference is, in some cases, the effect, the neurophysiological effect of dopamine binding is going to produce inhibition, and in some cases, it's going to produce excitation. Okay? So now this nigrostriatal tract, again, the vast amount of dopamine in the pars compacta is shooting up and is primarily dopaminergic. But I told you that 50% of the pars compacta substantia nigra are non-dopaminergic cells. Some of them do go up to the striatum, but the vast majority of the non-dopaminergic output of the pars compacta goes up into the thalamus. And when we do the thalamic nuclei, we're going to see the VA nucleus and BL nucleus of the thalamus is going to be receiving nigrothalamic input. And then we're going to see what's happening there. So now, that's the pars compacta. What about the pars reticulata? That's the second part of the substantia nigra. Whereas compacta, tight cells right next to each other, we have already learned that the word reticulata means net-like. So what you have a lot down here are fibers, okay? And some of the fibers that are coming in from the Raphe. They're coming in from the pedunculopontine tegmental area, cholinergic. They're coming in from the locus coeruleus and ventral noradrenergic, norepinephrine. But the other interesting thing is, whereas we have a nigrostriatal tract, we also have a strio-nigral tract, which is why that damn hyphen is very important. Remember, nigrostriatal is an ascending tract because you're going from the substantia nigra of the midbrain into the neostriatum of the telencephalon. The strio-nigral tract is going from the telencephalon down into the substantia nigra pars reticulata. Now, we pointed out that the nigrostriatal tract is 50% dopaminergic, more than 50% dopaminergic. It has the vast majority of the dopamine cells are going up to the striatum. So there's a dopamine signature there. The strio-nigral tract has also a very strong neurochemical signal. That strio-nigral tract is GABAergic. And so when you look at the neurotransmitter GABA and you're trying to do a free association between the word GABA and something else, the typical word that comes strongly associated with the word GABA is inhibition. Remember, glutamate is a classic excitatory neurotransmitter. In 99% of the cases, whenever there is GABA release, the activity on a postsynaptic cell is inhibitory, okay? So what you're basically doing there is you're producing powerful inhibition. And what we're going to learn is that there's a vast majority of GABAergic output coming out of the striatum, which is basically saying, stop right there. And again, that brings me up to the wonderful Meatloaf song, the second of the Meatloaf songs, Paradise by the Dashboard Light, which is basically a slightly older adolescent who actually ends up going out with the girl of his dreams. And she actually said yes on a date, and they're in the backseat of a car. Okay, so I'll just leave it there. And then there is a wonderful sound coming out of the woman singing in the song where she goes, stop right there. And I encourage you all to listen to the song and figure it all out. But the point is that GABA is basically saying stop things. And then what you need is the excitation to break through. So you now basically move to something that's a motor act and you're actually going to do it. So now let's look at the substantia nigra, pars compacta, and pars reticulata, and now look at it vis-a-vis dysfunction. And one thing that you can look at is a dysfunction that basically says we all of a sudden lose the capacity or the functional ability of the substantia nigra dopamine neurons to work. And the dysfunction that you see there is associated with the neurological disease, Parkinson's disease. Parkinson's disease doesn't afflict that many people. It obviously is afflicting more and more and more and more people as we age, because there's a stronger correlation towards having Parkinsonian symptoms as you age. Very often we study people who shouldn't be showing this sort of motor dysfunction earlier because of a loss of these dopamine nigral fibers. And the other interesting thing is you have a classic animal model of this. And that is work done by the Swedish neuroanatomist Urban Ungerstedt. When I pointed out the whole thing about histofluorescence, you had this work by Dahlstrom and Fuchs that identified norepinephrine-containing neurons, dopamine-containing neurons, serotonin-containing neurons. And basically, they basically said, well, here's where these cells are and where they sort of project. Ungerstedt, 7 years later, and this is like one of the last classics of all time that I always consider. There was a trio, the Dahlstrom and Fuchs paper, the Ungerstedt paper in 1971, and then a further paper by Lindvall and Bjorklund. That's the big trio of looking at the whole thing of catecholamines and indolamines in the brain and how they control. All of them were published in Acta Physiologica Scandinavica. And the way that they published things then was in exactly the sort of way things were classically published. So if you basically look at the work, the monumental work of Ramon y Cajal, Ramon y Cajal published his work in maybe something like 50 papers. Maybe he had more. But what they did was they were enormous papers. And the Dahlstrom and Fuchs paper, the Ungerstedt paper, and the Lindvall and Bjorklund paper were all published in single issues of Acta Physiologica Scandinavica. And Ungerstedt put these dopamine neurons, norepinephrine neurons, and serotonin neurons, and put them into classic neuroanatomical pathways. He united the neurochemistry with the neuroanatomy. But he didn't just stop there. The second part of his paper basically utilized a new neurotoxin at the time. So it's now 50 years old. And that neurotoxin is called 6-hydroxydopamine, or 6-OHDA. And what 6-hydroxydopamine does is it's a compound, which is a neurotoxin that gets grabbed by norepinephrine and dopamine transporters and gets reuptaken up into a nerve cell, okay? And then destroys the norepinephrine and dopamine cells. So it diminishes it. So what do you now all of a sudden have? You can have an animal model of Parkinson's disease by injecting 6-hydroxydopamine directly into particular areas. And that's exactly what Ungerstedt did in these initial studies, where he basically injected doses of 6-OHDA into the substantia nigra, sometimes unilaterally, and basically showed animals that had all of a sudden bizarre behaviors. So if you have an intact dopamine system and you have a non-intact dopamine system, the control is contralateral. So now, instead of moving straight ahead, if you had two normal substantia nigras, you now end up going into circling behavior contralateral to the side of the lesion. And he basically demonstrated this with this 6-hydroxydopamine model. So what is Parkinson's disease? In Parkinson's disease, what you have is a depletion of dopamine in the substantia nigra that then results in a loss of dopamine innervation in the neostriatum. And you're going to learn that the neostriatum is the massive interface between an overlying cortex, like the prefrontal gyrus, and other parts of the cortex feed through the caudate and putamen that then feeds through the globus pallidus to control what is called extrapyramidal motor symptoms. What's pyramidal motor symptoms? Pyramidal motor system has to do with fine movement, fine gesticulation. Extrapyramidal symptoms are symptoms that are more general. And the first of the most major of these general things is, of course, what do you see in Parkinson's disease? You see what is called tremors. So what is basically happening is the person has a continual sort of shaking. What the hell does that do to motor function? Remember, whenever we have the cerebellum and the motor pathways interacting, you basically have a very good idea as to where you are in space at any given moment, so that when you execute a movement, you know how far you should go, do whatever the hell you're doing. Now, if all of a sudden you're tremoring, well, you don't know where everything is in space. So now, all of a sudden, the movements become problematic. On top of the tremor, the other issue of Parkinson's disease, especially as it begins to progress, is inertial movement. Because remember, what the striatum is doing is finally basically saying, okay, start that movement and integrate it with everything else. Well, now you don't have dopamine anymore coming in. And now all of a sudden, the person has difficulty initiating a movement, to the point where in the latest stages of Parkinson's disease, the person can hardly move at all, okay? So this becomes very, very problematic. Also, what happens is that the substantia nigra, a small amount of it, does go into non-striatal types of function and will play a very important type of role in motivational behaviors. So one of the other things that you see as Parkinson's patients live longer and longer and longer, there's a greater incidence of psychiatric symptoms in Parkinson's, like hallucinations, etc. Okay, so that's Parkinson's disease, and you're able to go and study that. A descending pathway, as I said, is the striatonigral system, which is GABAergic. And a deficit of the striatonigral system starts to affect another group of patients that is usually smaller in number, but in some ways far more insidious than Parkinson's disease. And that problem is called Huntington's chorea. Huntington's chorea is very marked by a striatonigral path that dies, that GABAergic system starts to die. So now, instead of having trouble moving, yes, you do have the tremors and whatever, but the tremors is simply a lack of inhibition there. The Huntington's chorea is marked by choreiform movements, which are these sort of bizarre, exaggerated, uncontrollable types of movements that then when you try to actually execute a movement, you're not doing it well. It's almost like feeling like you have flippers rather than arms. And the person may have choreiform movements in the legs, in the arms, and the person has enormous difficulties. Second, the person then starts to suffer far more psychiatric and cognitive difficulties. And this onset is much more apparent in Huntington's chorea than it is in Parkinson's disease. So it's a deeper type of cognitive and psychiatric affliction, much more like what you would talk about with Alzheimer's disease in terms of its severity, whereas Parkinsonian dementia, Parkinsonian cognitive difficulties are a little bit more subtle, and they only end at the very end. But probably the most debilitating and horrible aspect of the disease is its onset. Whereas most Parkinsonian patients start to show symptoms in their 60s and 70s and 80s, with a very small number of people in their 40s and 50s, and then I'll talk about earlier with some other kind of interesting thing. People with Huntington's chorea very often start to show their symptoms in their 40s and into their 50s. And the outcome, the amount of time a person lives with Huntington's chorea is far shorter than what is with Parkinson's. So lots, just sort of like with Alzheimer's, when you basically say you have early Alzheimer's and you come to Alzheimer-induced death, usually that's a picture of like 8 to 10 years, sometimes longer with various treatments. With Huntington's, it can happen pretty quickly. And what basically happens is the person may die in 8 to 10 years, so they don't last to 60 or 70 years old. And what's so horrible about this? What's so horrible about this is exactly after the point in time that the person, in all probability, has engaged in reproductive sex and has basically produced progeny. So now we come to the worst part of it, that Huntington's chorea, unlike Parkinson's, there is somewhat of a genetic component, but not a complete one. In Huntington's chorea, it is far more genetic. So people are starting to, people pass it on to their progeny. And basically what happens is you have the probability of progeny, and then maybe 3, 4, 8 years later, you find out you have Huntington's, and now you have a fairly high probability that your progeny has Huntington's. And the person who basically studied this in an incredible way was a woman by the name of Nancy Wechsler, Wechsler, who she and her sister, both of their parents died of Huntington's. And she really set out to try to understand this. And what they did was actually find a natural population of people with Huntington's. And that is in some islands off of Venezuela, where you had some indigenous groups of people who basically lived in small groups in these islands that had a lot of intermarriage. You would have this enormous number of people with Huntington's disease. And with that, she basically, through the psychological study, the physical study, and then genetic studies, were then able to identify the exact gene that basically went wrong, and then was able to create some type of system by which you could then administer to a person and find out whether the person had Huntington's disease at a much earlier age, and then maybe make some decisions as to whether or not they should reproduce or whatever. And what was very interesting about the personal story is, even though she did all of this work, she demurred in trying to find out whether she had Huntington's or not. And her companion, a lifelong companion, is another person I hope that you all recognize, and that is Dr. Francis Collins, who is the current head of NIH. Okay, so there we have, very interestingly, within the substantia nigra, pars compacta, and pars reticulata, a system by which you have anatomically defined pathways that, when they show deficits, not only show deficits in behaviors in animal models, but also have an analog. And of course, one of the most famous people, well, two famous people that with Huntington's disease, of course, with Parkinson's disease, of course, was Muhammad Ali, probably the greatest heavyweight champion of all time. And then, of course, a guy who was a staple as a teenager in a show called Family Ties, named Michael J. Fox, who then went on to do a trio of movies called Back to the Future and a whole bunch of other movies, developed Parkinson's disease, and has been a tireless advocate for through the Michael J. Fox Foundation in terms of studying Parkinson's disease and treating it. So now this brings me into a full circle. So here I am talking to you about this. In 1980, there was a student in my neuroanatomy class who also joined my lab when he was a senior in college at Queens College when I arrived at Queens in 1979. His name is Jeffrey Kordower, Kordower. Jeff did a dissertation with me and finished all of his work between 1980 and graduated in early 1984. Jeff, when he was looking for postdocs, he had three major possibilities. One, we were doing analgesia work and whatever, and he could do a postdoc close to home in Rutgers University with a guy by the name of Barry Komisarek, with whom I collaborated, and he did a lot of analgesia work. The second was he would basically have to move from New York down to Atlanta, Georgia, and Emory University to do a postdoc with a guy by the name of Charlie Nemiroff at Emory. And Charlie, by then, was a wunderkind. He was in his early 40s, and he was already the chair of psychiatry at Emory and did lots of behavioral neuroendocrinology work. And the third place was to go work with these two guys named Don Gash and John Sladek up at University of Rochester. And so he was considering these things. And one day he comes into me and he goes, Rich, I don't want to disappoint you. And I go, what's the matter? What happened? He goes, I made my choice as to where I want to do my postdoc. I want to do my postdoc with Sladek and Gash. I'm assuming you want me to work with Barry Komisarek and still do analgesia research. Well, my feeling has always been that when you're doing a dissertation, yes, you can do exactly that type of work for the rest of your life, or you can view the dissertation as a means to an end and then go on to whatever the hell you really want to go and do. To which I said that, and he very happily moved up to Sladek and Gash, who was working in the area of neural transplantation and the early work of trying to figure out how you could physiologically manipulate in things like Parkinson's disease by placing in dopamine directly into the brain of a Parkinsonian model animal, and then basically see if you can restabilize this. Well, what he ended up doing was working with Sladek and Gash for about almost 3 years. And he really wanted to stay there. He said, oh, this is just a great lab. I'm going to go and continue and whatever. And of course, so this is a story, hopefully for all of you, that you can figure this out. So what Sladek and Gash said, well, the only way that you can come here is if you have an offer in your hand from somebody else. And then you can come back and then we can see whether or not we can fulfill that order up in Rochester. So Jeff goes out and he ends up going out to University of Illinois, Chicago Medical School, where Fred Plum is the head of neurology at University of Illinois Medical School. And there's a famous neurology textbook that's still famous to this day called Plum and Posner. Jerry Posner was at Sloan Kettering, Fred Plum at University of Illinois. They were some of the greatest neurologists of that time. Plum offers Jeff a position at University of Illinois, Chicago Medical School. So Jeff goes back to Sladek and Gash. Here's my offer, get me an assistant professorship. So of course they go and they check and they can't get him the position. There wasn't a position opening. So he has the choice of staying as a postdoc or going out with Fred Plum out into the University of Illinois, Chicago. He makes the decision to go out on his own. Thank God, because within 3 years, Sladek and Gash split up and they go to other different parts of the country, University of Kentucky, etc. So there is Cordauer out at University of Illinois, Chicago, where all of a sudden he's able to formulate a very, very strong primate lab to the study of Parkinson's disease. Within 3 years, he moves his academic appointment from University of Illinois, Chicago to Rush Presbyterian, probably the preeminent school, medical school in the Chicago area. Within 5 years after that, he becomes the head of the Center for Brain Repair at Rush Presbyterian. He still had his huge monkey colony back at the University of Illinois, Chicago, and he would work between the two of them. And basically, here is a kid from Queens with a psychology degree from CUNY, running not only the Center for Brain Repair and Neurology and Psychiatry and Neurosurgery, but also running a whole bunch of clinical trials with neurosurgeons underneath him. So how do we tie this all back together? During this whole time of his trajectory of 35 years at the University at Rush Presbyterian, he has always gotten NIH grants and whatever, and he also got support from the Michael J. Fox Foundation. So what did I find out from him a couple of months ago? He just got, in addition to 4 or 5 other NIH grants, he just got a grant from the Michael J. Fox Foundation for $8 million over 3 years to do all of the animal model and human model work related to Parkinson's disease. So you have your butts and seats right now, and I'm hoping all of the same kind of thing can go and happen to you in terms of making certain types of decisions. So now the last point that I want to cover in here is in here where what we saw was the interpeduncular nucleus. In here, we're seeing the ventral tegmental area. And I'm going to be talking about that in a little bit. So here we go. And here is our picture of the midbrain. So what you can see right up here is the superior, what we can see up here is the superior colliculus. We see the cerebral aqueduct. We see the periaqueductal gray. And then right in the ventral surface of the middle of the midbrain is the Edinger-Westphal nucleus and the oculomotor nuclei. Then right next to that, of course, is the MLF. And then what you can see, as shown by the letter M, and it looks like a whole bunch of teardrops. If you remember, we looked before when we were way back in the medulla and we could see the same thing. Remember when the gracile and cuneate nuclei form the internal arcuate fibers? What you can see very, very clearly here are the longitudinal tracts of the oculomotor radiations. Damn it, where is my pointer? I'm sorry. You can follow the letter M all the way down and see the oculomotor radiations. Then what you can see marked by the letter F, of course, is the red nucleus and the area around that. Then N, of course, is the substantia nigra. O is the cerebral peduncle, and P is the ventral tegmental area. So we started today by looking at the pons and the rostral pons and looking at the locus coeruleus and looking at it from the vantage point of norepinephrine containing pathways. What we will start in the next lecture is basically looking at the substantia nigra and the ventral tegmental area in terms of producing dopamine mesencephalic pathways. So let me come down here, stopping this and looking at our chats here. Okay, so unfortunately, I'll talk about the midbrain and go over the stroke question in the exam. I'll go over that next week. Okay, I hope that's okay with you. I don't think there's that many other messages right here. So does anybody have any further kinds of questions? Rocky Horror is the best Halloween movie ever. And people have never seen that. You can talk among yourselves. I think you want to basically say it's one of the funniest movies and bizarre movies at the time. And as you watch it in 2020, just think of the cultural aspects of things back around 1974 and how unbelievable, it's gender bending, it's everything in there, that sort of went on. And again, in my ridiculous free associations, I basically think when I talk about what I study, motivation, motor, all revved up with no place to go, paradise by the dashboard light, stop right there, whole things of behavioral inhibition based down to physiological inhibition. So if there is nothing else, I'm now at 12:01, so I don't want to go over. So we'll pick up here and do that. I'll try to grade the exams this week so people get feedback. The final 5 people, please show up tomorrow so we can finish that part of it. And then also in this week and next week, talk about when we want to do that makeup class. Okay, so with that, I have nothing else, and I'm going to close the meeting and have a very good week.